Hello everyone, I'm Dr. Divya. Today's topic for discussion is pentose phosphate pathway. The pentose phosphate pathway is a metabolic pathway parallel to glycolysis which generates NADPH and pentose, that is 5 carbon sugars, as well as ribose 5 phosphate. The pentose phosphate pathway is also called the phosphogluconate pathway or hexose monophosphate shunt. While it involves glucose oxidation, its primary role in anabolic rather than catabolic, it's an important pathway that generates precursor for nucleotide synthesis and is especially important in red blood cells. It involves the oxidation of glucose 6-phosphate to 6-phosphogluconic acid, which in turn is converted into pentose phosphates. In this pathway, glucose 6-phosphate is directly oxidized without entering glycolysis. Hence, it is also known as direct oxidation pathway. Reaction 1. 6 molecule of glucose 6-phosphate in the presence of coenzyme NADP are converted or oxidized into 6 molecule of 6-phosphogluconolactone by the enzyme 6-glucose 6-phosphate dehydrogenase. Here, 6 molecule of NADP are reduced in the reaction, which is reversible. Reaction 2. 6-phosphogluconolactone is hydrolyzed by the enzyme lactonase to produce 6 molecule of 6-phosphogluconic acid. Reaction 3. 6-phosphogluconic acid is oxidatively decarboxylated by the enzyme 6-phosphogluconic acid dehydrogenase. Here, 6 molecule of NADP are reduced, 6 molecule of carbon dioxide are released, and 6 molecule of ribulose 5-phosphate are produced. Reaction 4. 6 molecule of ribulose 5-phosphate isomerizes into 4 molecule of xylulose 5-phosphate and 2 molecule of ribose 5-phosphate in the presence of ribulose phosphate 3 epimerase and pentose phosphate isomerase respectively. Reaction 5. 2 molecule of xylulose 5-phosphate and 2 molecule of ribose 5-phosphate combine in the presence of trans-ketolase to form 2 molecule of pseudoheptulose 7-phosphate and 2 molecule of 3-phosphoglyceraldehyde. Reaction 6. 2 molecule of pseudoheptulose 7-phosphate and 2 molecule of 3-phosphoglyceraldehyde combine in the presence of trans-aldolase to form 2 molecule of fructose 6-phosphate and 2 molecule of erythrose 4-phosphate, that is 4 carbon sugar atom. Reaction 7. 2 molecule of erythrose 4-phosphate react with remaining 2 molecule of xylulose 5-phosphate in the presence of trans-ketolase to form 2 molecule of fructose 6-phosphate and 2 molecule of 3-phosphoglyceraldehyde. Reaction 8. 1 molecule of 3-phosphoglyceraldehyde isomerizes into dihydroxyacetone phosphate. The enzyme is phosphotriose isomerase. Reaction 9. Remaining 1 molecule of 3-phosphoglyceraldehyde unites with dihydroxyacetone phosphate in the presence of aldolase to form 1 molecule of fructose 1,6-bisphosphate. The later, in the presence of phosphatase, forms 1 molecule of fructose 6-phosphate. Reaction 10. 5 molecule of fructose 6-phosphates produced in reaction 6, 7 and 9 isomerizes into 5 molecule of glucose 6-phosphate in the presence of phosphohexose isomerase. The overall reaction of pentose phosphate pathway: 6 molecule of glucose 6-phosphate which enter into this pathway after oxidation produce 6 molecule of carbon dioxide, that is in reaction 3. Carbon dioxide come from carbon number 1 of the glucose molecule and 12 molecule of reduced coenzyme, that is NADPH2, which is involved in reaction 1 and 3, while 5 molecule of glucose phosphate are regenerated. 6 molecule of glucose 6-phosphate plus 12 molecule of NADP plus will give 5 molecule of glucose 6-phosphate plus 12 NADPH plus H plus along with 6 carbon dioxide. In other words, 1 molecule of glucose 6-phosphate after oxidation produces 6 molecule of carbon dioxide and 12 molecule of NADPH and H plus molecule. All the enzyme of pentose phosphate pathway are present in cytosol. Significance of the pentose phosphate pathway. The pentose phosphate pathway holds significant importance in cellular metabolism due to its crucial role in maintaining redox balances, protecting against oxidative stress, and supporting biosynthetic process. And this pathway significance is particularly evident in various cell types, which includes the erythrocytes. And these are all the references. Thank you.